All right, so we're going to talk about the influence of presynaptic receptors on neurotransmitter release. So first I'm going to draw our presynaptic terminal, and I'm not going to do much with the postsynaptic here because we already know what happens once neurotransmitter gets released. So presynaptically, our normal kind of easy situation is that we get our action potential come down. That action potential is going to hit voltage-gated calcium channels. And those voltage-gated calcium channels are going to be activated by the depolarization from the action potential. Calcium is going to come in down its concentration gradient. And then we're going to have it bind to already docked vesicles. And so we had our vesicles up here. We had them being filled with neurotransmitter. Then we had a little population of vesicles that wasn't docked yet. And then we had a population of vesicles that is docked. And it's docked there. It's attached to the cell membrane with what's called a snare complex. And those are just a whole bunch of proteins in a complex that just holds it right there, right next to the cell membrane, but not exactly touching it. And so those guys are just going to sit there, docked, until they get calcium bound. So this calcium that's coming in, yes, it's charged, but the charge isn't really the point. The point is it actually has to be the calcium ion itself that comes and binds directly to this snare complex. And once it's bound, what happens is these little guys change conformation, and they do this thing called zippering. And we can't draw that out, but you can imagine when you zipper, these guys are just kind of pulling the vesicle itself into conjunction with the cell membrane, which then allows exocytosis, and then it's going to allow neurotransmitter release. So the important thing to remember, even though a lot of times when you look at diagrams online about how calcium causes neurotransmitter release, they might show it coming in and binding to vesicles and sending them to the cell membrane, but that's actually not true. And the reason for that, if you think about it, how long do you want it to take to get neurotransmitter release after an action potential comes? The vesicles have to be already bound, they have to be already ready to go when the calcium comes in so that you get this instantaneous release. If the calcium was floating around, had to go find a vesicle, the vesicle then had to transport all the way down to the cell membrane, then the snare complex had to grab it and it had to come, the conformation had to come together before it would actually release, then that's adding a whole lot of time within the terminal between the action potential and release. So you've got to have it already docked with that snare protein. So this is kind of our normal standard neurotransmitter release that we think about. But one of the things about the nervous system is, of course, anything we say happens, it's always more complicated than that. So when we talk about neurotransmitter release and neuronal signaling, we usually have a little neuron here, and we show inputs coming in, an action potential coming down, and then release. And we usually show all of the inputs either coming into dendrites or coming into the soma. So those are other axon terminals. But what we don't usually talk about is the fact that we can have other neurons coming in and sending terminals onto the axon or onto the presynaptic terminal of neuron A. So we can have another neuron, neuron B, come in, and this is called an axoaxonic because the axon of this one is actually onto the axon of this one. And that's another way for another neuron or another circuit to come and change the activity of neuron A. So the things that are coming in here, the information coming in is going to tell us whether to have an action potential. But the information coming in here is actually telling us how much neurotransmitter to release and how likely we are going to be to release it in response to the action potential. So it gives us a little bit different information. Instead of this all or nothing action potential, now with the axoaxonic, we have this modulation. So how much are we going to release? Are we going to increase or decrease the sensitivity of this synapse? So that is one way to get another input from other neurons into this circuitry. And then another way to get input and alter how much release occurs is going to be if you have presynaptic receptors that are autoreceptors. And so these guys are receptors for the neurotransmitter that is being released. So it's a negative feedback mechanism. If you release a lot of neurotransmitter, then that is going to come around. It's going to activate receptors on the presynaptic terminal, and almost always that's going to be inhibitory and decrease the amount of new transmitter released. So basically it's just a way to keep from overdoing it, from over-releasing. So let's draw that in here. And one of the most common examples that you probably heard about are M2 receptors, muscarinic 2 receptors, or alpha adrenergic autoreceptors. So here we're going to have an autoreceptor. And these are almost always G protein. So we're going to have our little complex. And here what we're doing is that neurotransmitter is released. It's doing whatever it's doing down here. And then it's coming back up. And yeah, you're going to have your enzymatic breakdown. You're going to have your reuptake, whatever's going on within that synapse. But you're also going to have some neurotransmitter come over here, bind the autoreceptor, and then those G proteins are going to get released. And depending on what type of receptor this is, they're going to have different effects on the N-terminal machinery. So usually this is going to be a GIGO. That means it's our inhibitory. And when this happens, we're going to have all sorts of different downstream effects, different second messengers. One of the major things that happens is often cyclic AMP goes down, and a lot of things don't get phosphorylated or get dephosphorylated as a result. So let's draw another calcium channel over here, just so we have one on both sides here. That's, again, our voltage-gated calcium channel. And we're going to have this cyclic AMP and other types of second messenger pathways can come over here and phosphorylate this ion channel, or in other ways up or down regulate parts of the snare system. And in that way, if it's a GIGO and we're decreasing cyclic AMP, then what we can do is actually decrease the amount of calcium that comes in, or decrease the sensitivity of the snare complex, or any of a number of other things that will actually decrease the ability of an action potential to cause neurotransmitter release. So if this voltage-gated calcium channel is less active, then you're going to get your action potential come down, and it's still going to depolarize the terminal. But now this guy may not respond as much. And so if less calcium comes in, less calcium binds to the snare proteins, fewer vesicles are going to be released. So this is a way that you can still have release, because remember, the action potential is still coming, but you can decrease the amount of release once it gets there. So this is kind of our most basic system, where you have these autoreceptors. Now let's talk about those axoaxonic receptors. And those are ones that are coming in when we had up here with neuron B. Those were coming in from other neurons. And so I'm going to draw two different other neurons that could come in. So we've got one here. That's a funky dendrite. And a very big presynaptic terminal. And so say this is neuron B. And say it's an inhibitory pathway that's coming in. So here's our G protein. It's a GI-GO. And this could be GABA. It could be any other neurotransmitter that has an inhibitory postsynaptic receptor. So if neuron B wants to come in and modulate the amount of release with each action potential, then it's just doing the same thing that that autoreceptor did. It's coming in, it's releasing its neurotransmitter, and it's going to activate the GI-GO, which is then going to decrease the amount of cyclic AMP, which then goes and does all those other downstream things. So over here we have this axoaxonic, which we also call a heteroreceptor. And hetero means different, and homo means same. So it's a heteroreceptor, and axoaxonic just means where it goes. So one of the things to remember is autoreceptors down here have to be the same neurotransmitter that's being released because it's coming from that specific nerve terminal. Heteroreceptors can be any neurotransmitter. So we're talking about if, say, neuron A was a cholinergic neuron, then neuron B could be serotonergic. And so it's coming in and it's using a different neurotransmitter to modulate release here. So here on this side, we talked about, so then on this side, all of the effects over here we talked about were inhibitory to release. We had the autoreceptor inhibitory, and we had an inhibitory heteroreceptor. But you can also have facilitatory. And so if we go back over here to the left side now, we can have another neuron come in. An even uglier neuron. And that's neuron C, of course. And then it's got, say, another neurotransmitter, a third neurotransmitter. And it's coming in. And if it's activating, say, GS, then we're going to have downstream effects that are going to increase neurotransmitter release. So this GS can affect cyclic AMP by increasing it, and phosphorylate, increase phosphorylation of the calcium channel so you get more calcium coming in, and then you have more of a chance of calcium binding to those snare proteins. Or again, it has multiple different downstream effects that are possible depending on which G protein you have. So here on this side, we had those excitatory heteroreceptors. And those were going to increase neurotransmitter release probability or amount of neurotransmitter release, whereas the inhibitory neurotransmitters decreased amount or probability of release. So this looks pretty complex now that I'm looking at it, but hopefully we went through it cleanly enough that you can kind of separate down here. If I drew a little line down this terminal and you were looking at the way on this side that excitatory inputs affect it and the way on this side that inhibitory inputs affect it. So now a last thing that I want to mention here is that we have drugs that can go and act at these presynaptic inputs. So we can either use an antagonist that blocks that receptor, or we can use an agonist that activates it. By doing so, we can change the amount of neurotransmitter being released at that nerve terminal. And same thing over on the left side with excitatory heteroreceptors. We could put an agonist to that receptor or an antagonist, and we can change its input onto neurotransmitter release. So we're going to see that we have drugs that we're going to use either to decrease or increase neurotransmitter release at particular terminals. One thing that's really important to remember is we'll talk about examples where serotonin receptors influence dopamine release. So, for example, we have up here in this corner on the right-hand side, for example, we have a system in schizophrenia where the neuron A is a dopaminergic neuron and neuron B is a serotonergic neuron, and we can modulate the amount of dopamine being released by modulating serotonin receptors. We modulate presynaptic serotonin receptors here. So you'll stick around and we'll be able to talk about some of those. And hopefully this helps explain some of this extremely complicated looking presynaptic modulation.